by Chloe Rimkus, Marissa McCabe, Caitlin Rohr, and Catherine Romero.
The equation for photosynthesis is 6CO2 plus 6H2O plus light energy, which equals C6H12O6 plus 6O2. Plants get each reactant they need for photosynthesis from the environment. On the surface of the leaves of the plants, there are a large number of tiny pores known as stomata or stoma, and this is where the plant takes in carbon dioxide from the air. The plants suck up the water needed for photosynthesis from their roots. Chlorophyll captures energy from the sun. Chlorophyll is found in the thylakoid sacs of the chloroplast. The chloroplast is located throughout the cytoplasm of the cells of plant leaves. These reactants are later used to produce glucose and oxygen. There are two different types of reactions that have to take place inside of the chloroplast in order for this to happen. The first reaction is light dependent, which means that the plant depends on the sun to make the water molecules split apart. The chloroplast takes NADP and ADP and a phosphate to produce ATP and NADPH. Then the carrier molecules store the hydrogen produced for future use to make glucose for the plant. The second reaction is called the Calvin cycle, and it takes carbon dioxide, ATP, and NADPH to produce sugar, NADP, and ADP. These two reactions form a large cycle where the plant takes ADP, which is a non-usable energy, to produce ATP, which is energy in the form the plant can use.
We believe that our altered variable, using room temperature spinach as opposed to our control group of refrigerated spinach, will increase the rate of photosynthesis. This is because the molecules are interacting faster and ATP is being pumped faster, therefore speeding up the process of photosynthesis.
There are 10 leaf discs in our syringe, and we're going to suck up a little bit of 0.2% bicarbonate solution.
Still breathing hard, and they're like, so.
Okay. The materials we used were an incandescent lamp, a beaker, a petri dish, distilled water, 0.2% sodium bicarbonate water, a disposable pipette, a timer, soap, tweezers, a syringe, a hole punch, and spinach, both refrigerated and room temperature. The independent variable in our experiment was the temperature of the spinach, and the dependent variable is the number of the spinach discs that floated within a 20-minute interval, which represents the rate of photosynthesis. This dependent variable was impacted by whether we were using room temperature spinach or refrigerated spinach. The constants were the amount and concentration of sodium bicarbonate solution, the incandescent light used, the size of the leaf discs, the amount of time measured, and the use of the amount of distilled water as a heat sink.
Comparing the control group data to the experimental group data, the rate of photosynthesis did increase with the use of room temperature spinach. This relates back to our hypothesis because we predicted that our altered variable, using room temperature spinach as opposed to our control group of refrigerated spinach, will increase the rate of photosynthesis. The higher the temperature, then typically the greater the rate of reaction of photosynthesis. Photosynthesis is a chemical reaction, and the rate of most chemical reactions increases with temperature. Increasing the temperature makes the molecules move and collide faster, increasing the rate of the reaction.
The purpose of creating a vacuum with the syringe was to suck out all the air from the leaves, preventing them from performing photosynthesis. Removing all the air from the leaves also allowed for them to sink to the bottom of the beaker. The part of the photosynthesis equation we altered was the amount of carbon dioxide in the leaf discs, since we initially removed all of it with the syringe and the source of light, as we use an incandescent bulb and not natural sunlight, which is the normal light used in natural photosynthesis. The leaf discs got the carbon dioxide to perform photosynthesis from the bicarbonate solution they were placed in. The leaf discs float because they are producing oxygen while they perform photosynthesis, and the oxygen allows them to float.
One source of error within our group was placing the leaf discs in a beaker by itself without the bicarbonate solution for the first minute. However, we fixed this quickly by transferring it into the correct container. Another source of error was some light from the window was peeking out near our beaker. This could have made the leaves begin to perform photosynthesis before we began collecting our data. In the control experiment, we used a drop of soap, and we did not do the same in our experimental group.
This group tripled the concentration of the sodium bicarbonate solution. They went from 0.2% for their control group to 0.6% for their experimental group. Throughout the control group, none of their leaf discs floated. However, after they tripled the concentration of the sodium bicarbonate solution used, all 10 leaf discs floated within a 10-minute interval. Their independent variable was the concentration of the sodium bicarbonate solution, and their dependent variable was the number of spinach discs that floated within the 10-minute interval.